Hi everyone. I am going to summarize chapter 15 lessons and assessment questions. We are also going to solve them. So of course for our objectives, we're going to summarize and explain the main points of the lesson and solve the assessment questions that are given under each lesson. So let's first summarize 15.2 recombinant DNA. In copying DNA, genetic engineers can transfer a gene from one organism to another to achieve a goal, but first, individual genes must be identified and separated from DNA. The original method, which is used by Douglas Brashear, involved several steps. Use a complementary base sequence to attract the predicted mRNA. Predict the mRNA code for that sequence. Determine the amino acid sequence in a protein, and find the DNA fragment that binds to the mRNA. Once scientists find a gene, they can use a technique called the polymerase chain reaction to make use or to make many copies. First, heat separates the DNA into strands. As the DNA cools, primers are added to opposite ends of the strands. Then DNA polymerase adds nucleotides between the primers, producing two complementary strands. The process can be repeated as many times as needed. In changing DNA, so recombinant DNA molecules continue DNA from or contain DNA from two different sources. Recombinant DNA technology can change the genetic composition of living organisms. Plasmids are circular DNA molecules found in bacteria and yeast. They are widely used by scientists studying recombinant DNA because DNA joined to a plasmid that can be replicated. A genetic marker is a gene that is used to differentiate a cell that carries a recombinant plasmid from those that do not. In transgenic organisms contain genes from other species. They result from the insertion of recombinant DNA into the genome of the host organism. A clone is a member of a population of genetically identical cells. So for lesson 3, the applications of genetic engineering. Genetic engineers work to improve the products we get from plants and animals. Genetically modified crops may be more nutritious or higher yielding. So that means they may be resistant to insects, disease of spoilage, and some can produce plastics. Genetically modified animals may produce more milk, have leaner meat, or contain higher levels of nutritious compounds. So transgenic salmon grow rapidly in captivity. Transgenic goats also may produce spider silk in their milk. In health and medicine, recombinant DNA studies are leading to advances in the prevention and treatment of disease. Examples include vitamin-rich rice, human proteins made in animals model of human disease for research, and bacteria that produce human insulin. Gene therapy is the process of changing a gene to treat a disorder. However, gene therapy is still an experimental and had high-risk technique. Genetic testing can identify hundreds of inherited disorders. Not all genes are active in every cell, so DNA microarray technology lets scientists study thousands of genes at once to determine their activity level. In personal identification as an application of genetic engineering, DNA fingerprinting analyzes sections of DNA that may have little or no function but that vary from one individual to another. DNA fingerprinting is also used in forensics, that is the scientific study of crime scene evidence, to identify criminals. It is also used to identify the biological father when or paternity is in question. Common ancestry can sometimes be determined also by using mitochondrial DNA and Y chromosome analysis. In Lesson 4, the ethics and impacts of biotechnology, most of the research in genetic engineering is done by private companies. They patent their findings and inventions to protect their investment and make a profit. The patents block other scientists from pursuing certain lines of research. In 2007, the Genetic Information and Discrimination Act was signed into law in the United States. It prohibits discrimination based on genetic information. In safety of transgenics, there is controversy about the safety of GM foods. Prominent genetically modified foods argue that GM crops are better, safer, and higher yielding than conventional crops. GM crops require less land and energy to grow, and insecticides need not be applied to insect-resistant strains. Careful studies have provided no support to concern about the safety of GM crops. Opponents or opponents argue that the safety of GM crops has been neither adequately tested for long-term use nor regulated, so patent of GM seeds may force small farmers out of business. The resistance of GM plants to insects may harm also beneficial insect species, while resistance to herbicides may also result in the overuse of toxic chemicals. Some states have introduced legislation to require that GM foods must be labeled to give consumers the chance or the choice. In ethics of new biology, few have argued that gene therapy for curing disease is ethically wrong, but many ask questions of how far genetic modification should go. Is it right to try to engineer children to have certain characteristics? Or should human cloning be allowed? So let's have now some assessment questions. Let's start with 15.2, which is found on page 442. Let us just answer a few questions from it. So let's have number 13 question on page 442. Describe what happens during a polymerase chain reaction. Of course, the first step is to heat a piece of DNA, which separates into two strands. Then, as the DNA cools, primers bind to the single strands. Next, DNA polymerase starts copying the region between the primers, and these copies can serve as templates to make still more copies. For our next question, number 14. Explain what genetic markers are and describe how scientists use them. Can you guess the answer? Okay, for the answer, a genetic marker is a gene that makes it possible to distinguish bacteria that carry a plasmid from those that do not. Genetic markers are also inserted into plasmids so that scientists can identify cells that have been transformed. Let's have now Lesson 3 assessment questions in page 443 of your textbook. Let's have number 21 question. Describe how a DNA microarray might be used to distinguish normal cells from cancer cells. Okay, try to guess the answer, or what do you think is the answer? Okay, for the answer, DNA microarrays allow scientists to compare the gene expression patterns of different cells. This kind of comparison can be used to distinguish cancer cells from normal cells. Let's have number 22 question. Describe 2 important uses for DNA fingerprinting. For the answer, DNA fingerprinting can be used to help solve crimes by matching DNA evidence from a crime scene with possible suspects. It can also be used to establish familial relationships, such as in cases of disputed paternity, by finding alleles in a child that are not carried by the mother and then matching them to the father's fingerprints. Okay, for our last lesson, lesson 4, assessment questions, page 444. Let's have number 26 question. Give an example of a disadvantage associated with patenting genes. For the answer, sometimes patent holders demand high fees that block other scientists from exploring lines of research. Number 27 question. What is one argument used to critics or by critics of genetically modified foods? For a sample answer, expensive GM seeds may force small farmers out of business, especially in the developing world. So that's it, girls. Thank you for watching. Till next time.